Trisha Brandon. I'm the Program Director here at Town Hall Seattle, and it's my pleasure to welcome you to this evening's panel discussion on cutting-edge brain research. Before we get started, I'd like to mention a few other science, civic, and arts and cultures events that are coming soon to Town Hall. We have on April 4th, John Seisel with a new holistic philosophy of Alzheimer's care. Then on April 7th, the Massachusetts nonprofit National Priorities Project with its Empowering a People's Guide to the Federal Budget. And then on April 15th, we welcome Iraq War veteran Brian Kastner with a look at the dual realities of war and the life that follows. This is just a taste of the next few weeks at Town Hall, and the best way to keep up to date with our programming is to become a member. Membership starts at just $35, offering downfront seating, discounts on book and ticket purchases, and a mailed copy of our monthly calendar. And most importantly, it provides the support essential to bringing you more than 400 events annually at affordable ticket prices. So if you believe in Town Hall, please become a member. You can find out more at our website, townhallseattle.org. Tonight's panel is presented by the Institute for Systems Biology and Town Hall as part of the Seattle Science Lectures, sponsored by Microsoft, and with series media sponsorship provided by KPLU. Our panelists will speak for about 40 minutes, after which they'll take your questions. We'd ask that you come to the mics that are at the edge of the stages, here and there, so that we can pick you up for the recording that we're making. Now our panelists. The moderator of tonight's discussion is Dr. Nino Ramirez, Director of the Center for Integrative Brain Research at Seattle Children's Institute, Professor of Neurological Surgery at the University of Washington, and a research affiliate at the Center on Human Development and Disability. He has a general research interest in the neural control of rhythmic and studies neural mechanisms involved in the generation of respiratory rhythms, neocortical activity, and epilepsy. And then our panelists include U.S. Army retired General Peter Chiarelli, who was appointed Chief Executive Officer of One Mind for Research in 2012 after retiring from the Army as a 4-star general with 40 years experience. As the 32nd Vice Chief of Staff in the Army, Chiarelli was responsible for the day-to-day operations of the Army, including extensive involvement in its behavioral health programs. As the CEO of One Mind for Research, an independent nonprofit that seeks to rapidly develop new treatments and cures for illnesses and disorders, Chiarelli continues his advocacy for eliminating the stigma associated with the invisible wounds of war, post-traumatic stress, and traumatic brain injury. Additionally, we have panelist Dr. Christoph Koch, who is the Chief Scientific Officer at the Allen Institute for Brain Science, which he joined in 2011. For the past 25 years, Koch has served on the faculty at the California Institute of Technology, most recently as the Lois and Victor Trundle Professor of Cognitive and Behavioral Biology. Previously, he spent 4 years as a postdoctoral fellow in the Artificial Intelligence Laboratory and the Brain and Cognitive Sciences Department at the Massachusetts Institute of Technology. Koch has published extensively and is the author of the book The Quest for Consciousness, a Neurobiological Approach. And finally, Dr. Leroy Hood is the pioneering president and co-founder of the Institute for Systems Biology, who with colleagues at Caltech developed 4 instruments that paved the way for the successful mapping of the human genome. Dr. Hood has played a role in founding more than 14 biotechnology companies, including Amgen and Darwin, and is a member of the National Academy of Sciences, the National Academy of Engineering, and the Institute of Medicine. He's co-authored numerous textbooks, as well as a popular book on the Human Genome Project, The Code of Codes, and received the 2011 National Medal of Science. Please offer your warmest welcome for our distinguished guests.
Thank you very much for coming here, and we're really honored. But I think you are in for a treat because these are like 3 legends in their fields, and I thought I had the honor to introduce them, and I biked here, but I totally destroyed my carbon footprint because I accidentally printed the resume of Dr. Leroy Hood. 700 publications and I don't know, 20 pages of awards, and so we decided. So I think I won't do this because otherwise we spend most of the time introducing each other. And so I hope that this brief introduction of everybody gives you a little bit of an overview of what we're about, for our backgrounds. And I think the beauty about these backgrounds is that we come from different disciplines. Like we have genome sciences, we have cognitive sciences, and then we have military background. And Dr. Chiarelli played a big role in brain research insofar as he was instrumental in recognizing post-traumatic stress as a major problem in soldiers, and he's very much involved in initiatives to actually address this issue in a big way. And so what we decided is that we will basically do two blocks. The first block will be about big sciences, and the second block will be how do we demystify the brain, because the title was Demystification. And but we also decided that we don't go into much detail. We want to give you an overview. Everybody gets like 5 minutes only, and for each aspect. But then we would like to open it up for the question and answers, because I think everybody is fascinated by the brain. Everybody comes with a big load of questions, and I think in this dialogue we might be able to address these big issues and how we go about understanding the brain. And I think it's also a very good point to start this town hall meeting, because President Obama said last night, as humans we can identify galaxies light years away, we can study particles smaller than an atom, but we still haven't unlocked the mystery of the 3 pounds of matter that sits between our ears. And this is really something that is really fascinating. Why is this piece of tissue such a complicated beast? And we have like 100 trillion 100 trillion cells in our body, but in the brain we have 100 billion. So it's not that big compared to the overall picture, but the major difference of the brain compared to any other organ is that in the brain it really matters what every single cell is doing, how they're connected with each other, how they talk to each other. So if I tell you something right now, I have to activate very, very specific neurons in a very timely fashion to tell you something. And even though I do this in a very precise manner, you still probably hear that I have a German accent. And the reason is that I have a German accent is because my motor control I couldn't learn specifically enough to mimic exactly those sounds. So this is the specificity that we have to encounter, and so how do we unravel this and understand consciousness? And so these are the issues that we would like to address today. And coming to the first block, the big science block, maybe Christoph, do you want to lead on with the first announcement?
Thanks a lot, Nino. Yeah, as you heard Nino state, yesterday President announced a new initiative, a new federal initiative in the presence of 200 neuroscientists. There haven't been so many neuroscientists at the White House, which by it's a wonderful thing. So it's mainly for the development of new technologies. This the first year it will be funded by a relatively modest $100 million investment. But the idea is that this is a down payment on something much larger. Of course, it still has to be approved by Congress, who has proven reluctant to fund some of these ideas. This has to be viewed on the background of sort of in general big science or neuroscience. The field that we represent is sort of traditionally a field of small lab science, where typically you have a professor and her 2 postdocs and 1 or 2 grad students doing most of the work. They are on the order of 10,000 labs worldwide, 10,000 labs that study the brain, and maybe 50 to 60,000 neuroscientists worldwide. So they are existing over the last year. There have been 2 very large projects that have been announced that study the brain in what's called a big science fashion, which we're used to in physics. You may have heard of CERN, the nuclear lab in Europe, in Geneva, that found the Higgs boson, or the large telescopes that we're used to that sit on Mount Kea. Those are all projects that involve thousands of scientists and very large budgets. So there was one announced by our very own Paul Allen here in Seattle last year at the Allen Institute for Brain Science that I represent, and he endowed it with $300 million for the first couple of years, plus he also recently gave us a building here in the Lake Union area, and that's meant to study the functioning of cortex, which is really the heart of what makes us human, which makes us think and remember, and also that makes us conscious both in animals, primarily in the mouse, as well as in humans. Then there's a second effort that was recently funded by the European Union. It's a large effort. It's over 10 years. It's roughly $1.4 billion. It's called the Human Brain Project. And it's mainly meant to provide computational infrastructure, to provide computers that can simulate the brain, the human brain. But again, everybody talks about the human brain, but by and large, most of the research involves animal brains, like mice, for instance. Mice they look very different from us, but if you look at their brain, it's actually remarkably similar. And if I give you a little pea, a little sort of tiny sized chunk of mouse brain and tiny sized chunk of human brain, very few people can tell the difference, because by and large, the brain tissue in all mammals is very similar. The difference is we have roughly 1,000 times more than a mouse. So these are the two large existing efforts to which now the U.S. federal effort will be joined.
I think that the idea to have big science around neuroscience is really a very important idea, and because it really brings people together with different disciplines to address one question. But I think we can learn a lot from prior big projects, and because the brain science projects are not the first. And Dr. Lee Hood is actually one of the leaders in genome sciences, and he was instrumental in this whole genome project. And I think if Lee can tell us something about this, I think it will be very inspiring and give us some lesson for neuroscience.
The Human Genome Project really got started in 1985 amid enormous skepticism. 90% of the biologists were against it. The National Institutes of Health was against it. And it was basically about the central issue of what it was to do, namely to decipher the 3 billion nucleotides present in the human genome. There was skepticism, A, about whether it could be done, B, about, if done, that it would be worthwhile, and C, it was viewed as a repetitive task, and it was believed that you couldn't get very good scientists to do this. So there were lots of arguments against it. It did, in fact, get started in 1990 and finished 13 years later. It was very instructive because, on the one hand, the Human Genome Project differs in one very fundamental way from the proposed Brain Project. And that fundamental way is the Human Genome Project was very precisely defined and circumscribed. We just wanted to get the order of all the different letters of the DNA language in your genome. The Human Brain Project is, by contrast, an enormous project in dimensionality, because in many cases we don't even know today quite what questions to ask. But in other ways, the Human Genome Project was, I think, going to be a parallel for what the Brain Project, if it's successful, could really be. So one, the Human Genome Project was perhaps the first real example of big science. And it demonstrated the power of what big science could do. Number two, it actually created a parts list that told us all the genes, and by inference, all the proteins that existed in humans, and it opened up this new field called systems biology, because you needed the parts list before you could study the complexity of the human being. Number three, it democratized the genome. It made every part of the genome available to any biologist that was interested in studying it. And it went on to revolutionize the genomes of plants and animals and microbes. It started important phases in medicine. It raised really interesting ethical questions. 5% of the Human Genome Project was spent on looking at the social, ethical, and legal questions of the Genome Project. In many ways, it was seminal because it brought to biology computational people, and they stayed because they had something to do for the first time. It created open source attitude. It made all the data immediately accessible. These are all fundamental components of contemporary science, and hopefully the Brain Project will do exactly the same thing. But I will say, an estimate that some have argued about, but it really is a fascinating estimate, is it's had an enormous economic impact. So The Human Genome Project, its estimated cost, $3.5 billion over 13 years or so, and it's estimated that it has generated $800 billion worth of profit. So the return on investment is utterly staggering, and it's opened up whole new industries and whole new branches of modern medicine and so forth. And I suspect in many of these things that the Human Genome Project has done, we'll see beautiful parallels for the Brain Project.
Thank you very much. I think talking about the economics of this, coming to neurological disorders, I mean, we can expect a similar return. I mean, worldwide there are like 1 billion people with neurological disorders. I mean, obesity, which I consider part of a brain disorder because how do you control your appetite and saturation, is in the rise. Autism is in the rise. And major depressions, epilepsy, everything is really a big burden. And it's not only a big burden for the individual, but it's a huge burden for the economy. And if you think about the age curve, I mean, Alzheimer will be a major economic burden for us as we grow older. So clearly, I mean, investing in neuroscience will be a major step forward. And I think the genome sciences gives us optimism that actually the dollars we invest are returned, multiplied. So what I would like to do now is the next major really project that led the way actually in big sciences, and this is the Manhattan Project. And maybe General Chiarelli can talk about this a little bit.
Well, that's putting the general in the box, making the general talk about the Manhattan Project. That's really not fair. Let's try to moonshot.
Okay, go for it.
I need to make the first correction of the evening. I'm not a doctor. I'm a political science major who went to the Graduate School of Public Affairs here at the University of Washington and spent 40 years in the Army. I spent 2 tours in Iraq and had no idea what traumatic brain injury or post-traumatic stress was. And that's why I'm here tonight. When I became the Vice Chief of Staff of the Army, I was responsible for 1.1 million men and women. And what I thought were the most prolific wounds coming out of the war were soldiers who had been shot and lost an arm, a leg, or multiple limbs. That, in fact, was 10% of the total number of our most seriously wounded in 2008. And it has stayed that constant through time. When I became vice in 2008, 37% had traumatic brain injury and post-traumatic stress, and when I left, that number had increased to 67%. So by far, the most prolific wounds coming out of these wars are traumatic brain injury and post-traumatic stress, and that's why I'm here tonight. Having served 2 tours in Iraq and Afghanistan, I had no idea what they were. And I became frustrated in the 4 years I was the vice chief to see how much we didn't know. This isn't really something where we know a lot. It's really something where we know very little. There's 27 different definitions of traumatic brain injury according to the CDC. We've underinvested in this area for many, many years. Last year, the CDC estimates that $78 billion was spent in our medical system on people with traumatic injury to the head. And we invested a total of $84 million in trying to find out as much as we could about traumatic brain injury. Now, I submit to you, for those of you who are in business, if you have a $78 billion program and your R&D budget is $84 million, you're probably never going to solve the problem. And that's basically what I've seen. What one mind is trying to do is to fund the big science. We're saying the government can't do it alone. It's going to have to be a combination of public-private partnerships that come together that include philanthropy. You've got, I believe, the greatest example of that here in the city of Seattle, and I say that unabashedly, in the Allen Institute, an organization that is doing the work that it is doing based on a lot of private philanthropy and going ahead and publishing the data that they collect almost weekly online for scientists everywhere to use, and they are using it. It is an amazing thing. But I believe after looking at this that we're going to have to spend much more than $100 million. There is a need here for really big science, for putting together the kind of databases in a time when we can do things with data and imagery that we have never ever been able to do before. And what Dr. Hood has given us with the Genome Project allows us to understand some of the genetic makeup of these diseases. One mind is concerned about all the diseases of the brain, but we've decided to focus initially on traumatic brain injury and post-traumatic stress. And although I came at it from a soldier standpoint, the problem is huge. 3.4 million Americans every single year suffer traumatic brain injury. It could be a bicycle accident, could be a car accident, could be a sporting accident, could be falling off a ladder, you name it. And it's estimated that 8% of the total population will have post-traumatic stress in some time during their life. That's kind of an amazing figure when you realize we really don't even have good diagnostics for it today. So my belief is that if there was ever an area that was in need of big science, call it a Manhattan Project, but for tonight I'm going to call it a moonshot. We need to move from outer space to inner space and look at big science to get at these problems. Because I think the doctors will confirm that we're seeing more and more the connection of these different diseases. We tend to think of them as stovepipe diseases. In other words, if someone you know has Alzheimer's, you may invest in Alzheimer's research, but there seems to be some connections possibly between Alzheimer's, dementia, Parkinson's, ALS, traumatic brain injury. And it's only when we share the data and do this big science, I think we're going to get at these scourges.
Wow, that was impressive. Thank you. No, I think the crisis is acute. And, I mean, every day, children are subject to traumatic brain injury. It's actually children are very much affected. And actually, all sports, like kids playing sports, are as in danger as the professional football players. And what do we have to diagnose a traumatic brain injury? We have nothing. I mean, we have nothing is wrong, but we're doing neurocognitive tests, like asking, what's the score? Remember, like an hour, 10 minutes ago, who was the player who scored, etc. These are the approaches that we use, but they're like 50 years old. And what we should be able to do is have certain biomarkers that tell us exactly how serious is the injury and what are the consequences, because once we know how to quantify a traumatic brain injury, we will be able to actually much better predict the outcomes. And at the moment, it's really sometimes astonishing. I mean, even moderate or mild traumatic brain injury in some girls can lead to headaches for the rest of their lives. So it's really a very, very acute problem, and I think Pete is totally right to point out that this should be a major, major priority. So clearly need for big sciences, and what I would like to do now is move to the next part, and the mystification of the brain. And really, it's an important problem, because everybody knows that the brain controls our emotions, controls our thoughts, our cognitive behavior, our actions. Is it at all possible to address these questions? Can we demystify the brain? And I think Christoph leads the way in tackling, I think, to me, the biggest mystery in this consciousness. So Christoph, if you want to tell us how we go about it to understand consciousness.
3 minutes, I'll explain it.
What's first to come, huh?
For me, $5. So consciousness is the oldest problem in the panoply of problems that philosophers have addressed. It's the heart of the mind-body problem. Well, what is it that gives rise to these subjective feelings in our head? I have a picture right now of you, and you guys have a picture of me. How does this picture emerge? I can hear voices, I can remember things. This entire inner life that is our life. The only way we experience anything, in fact, to experience something needs to have a conscious experience. So there has been the focus of philosophers. Over the last 200 years, it shifted from metaphysics and philosophy into the clinic and into neuroscience lab. What we've realized over the last 200 years, just like to understand chemistry, We need to understand atoms and the way they interact, so electrons and outer electrons and shells and valence electrons, etc., like that. And what we've realized over the last 50 years, that it's also true for the brain, that in order to understand consciousness or memory or behavior or any of its pathologies and these diseases, we really need to look at the atoms of perception and the atoms of memory. Those atoms are nerve cells. There are a lot of nerve cells. Mentioned only the number 100 billion, so it's roughly there's many stars in the Milky Way galaxy as there are neurons in the typical brain. The trouble is these neurons are enormously complex. They're roughly there may be up to 1,000 different types of them. So it's not just that they are as a goo, it's a homogeneous goo. No, if you remember the Lego blocks, playing with Lego blocks, well, it turns out now you have a set of probably 1,000 different types of different colors, and the roof tiles and the flat ones and the 2x4 and the 1x8, and they all fit together in different ways. And we right now have very little ideas how they fit together. And so the problem we're facing is trying to explain something as complicated as consciousness, what I see, which is now being studied by many labs, or to explain pathology, like if you're depressed or you're schizophrenic, how to do that. Now, right now, the only way to do it, you go to a psychiatrist, and the psychiatrist gives you a questionnaire, and we use this sort of the same technique that we've used over the last 100 years. He asks you how do you feel, and you feel depressed. And trying to fix it, we sometimes prescribe things like Prozac, etc. That's a little bit like going to it's very crude. It's a little bit like going to a mechanic, and you tell the mechanic, well, my car doesn't work, and the only thing the mechanic does, he opens the hood and pours oil over it. And of course, sometimes some of the oil will go where it belongs, but also, the oil goes everywhere else and causes all sorts of side effects. So the emphasis over the last 30 years has been to study consciousness or brain disease at the level of neurons. And there we're making enormous progress, where we can now begin to map them at a very fine scale, so we can use all sorts of fancy techniques, mainly in animals, but also a little bit in humans, to look at this finely woven lace. It's a lace of incredible, it's beauty, it's very tenuous, because each of these tiny neurons is connected with 10,000 other nerve cells, very different from a computer. But we know enough already that the final answer is going to be enormously complex. And what people have done, they use a magnetic brain scanner, they use microelectrodes that sometimes people like epileptic patients have to have inserted for medical reasons, or they sometimes use animal research to study animals when they see. And so we already now know that something as specific as consciousness emerges from specific parts of our brain, from specific neurons that interact in very specific ways. They're not all over, they're in particular parts of the brain. And we also know this from books like you might have read, Oliver Sacks. Right? For example, the man who took his wife for his head. When we know you have a specific lesion, you lose a part of your brain, you have a little stroke or gunshot or something, then you may lose a very specific aspect of consciousness. And so by slowly encircling the brain and by slowly beginning to understand it and probe it, we begin to understand that consciousness is something that we can ultimately understand. It's not a problem that will forever sort of escape the ken of science, but ultimately we'll understand it. Now that's not only important to understand how we got here and what is the position of human in the rest of the universe, and which other entities are conscious, to what extent is a dog conscious, to what extent is a fly conscious, to what extent are all biological creatures conscious, to what extent is a fetus conscious, to what extent is an anesthetized patient conscious, to what extent, very important practical problem, is a patient who was in a severe car accident and all she can just sit there and moan, to what extent is she conscious. It's a so-called vegetative state. And people are developing tools to actually sort of develop a consciousness so we can measure this in clinical practice. And so, as I said, this is not only important to understand our place in the universe, but also for immense practical problems. In particular, for studying all the pathologies of consciousness, such as schizophrenia, such as depression, such as Alzheimer's, and all these other psychiatric diseases. So we're making progress, but these problems are very, very difficult. The brain is by far the most complex system in the known universe. So although we make progress, there will not be any answers in the next 1 or 2 years. It's going to take, it's going to take many years to fully understand and to eliminate these diseases.
Thank you very much, Christoph. And I think if we want to understand the brain, we have to really address it at different levels of integration. And Christoph is really a pioneer in addressing this big question at the level of the cortex. But big questions are, what can we learn even from other systems that are connected with the brain? And Dr. Lee Hood will tell us something about how can we learn something about the brain by looking at the blood.
So I remember thinking maybe 8 or 9 years ago about brain diseases and asking the question, how can we best study them? One way to study them, of course, is to reach in and take a piece of the brain and study it, and that isn't very practical with humans. So what we started thinking about is this view that in principle, the blood can be a window into health and disease for every single organ. And the idea that we had is in a diseased organ like the brain, there are biological networks that become disease-perturbed, and that the proteins that they control are altered in their levels. So what we identified was a fascinating series of proteins in the brain that are only expressed in the brain. When these are secreted into the blood, as we've identified almost 200 of them are, in the blood you can quantify them, and you can see how they change when disease occurs. So all you have to do to view how the brain is changing is take blood longitudinally at different points across a disease. And we've developed both the technologies for identifying these brain-specific blood proteins. We've developed the technologies for quantifying them very precisely in the blood. And really what they've enabled us to do are several spectacular things. Our first studies were to look in the mouse, because we can manipulate that experimentally very effectively, at an induced neurologic disease. It was a disease induced by infectious prions. It lasted about 22 weeks. We could gauge just exactly what was happening with that disease by measuring from the blood of the mouse these brain-specific blood proteins. And we were able to show that these proteins, as diagnostic markers, could do 4 striking things. Number 1, they diagnosed the disease much earlier than the clinical signs would ever let you do it. Number 2, they beautifully followed the progression of the disease. They could map exactly which disease-perturbed networks were brought into focus as the disease progressed. Number 3, we were able to stratify neurodegenerative diseases by these blood markers, because different neurodegenerative diseases change different combinations of proteins. And then 4, we haven't done this, but we are going to do it very soon in the future, is we'll be able to use these markers to follow what therapy does to the disease in the brain. And we've recently applied this to a human disease, post-traumatic stress disorder. So we recently studied 50 soldiers from Afghanistan who were normal, and 50 that had extreme forms of PTSD. And we were able to put together a panel of 8 biomarkers that had an incredibly high sensitivity and specificity for distinguishing these 2 populations, one from another. These are in the early discovery phase, and what we need to do now is move to a validation phase, and with Pete, we're going to be getting the resources to do that kind of thing in time. But the quality of these diagnostics is better than any I've seen in other studies that we make on many different diseases now. And it opens up an entirely new world of studying neuropsychiatric diseases. So 1, we're just getting ready to start studying traumatic brain injury, and I'm utterly convinced exactly the same approach will give us powerful diagnostics. And I'm even more convinced that the 2 types of diseases will overlap in absolutely fascinating ways. But even more important, I think for the first time we have a quantitative measure in the blood of what is happening in the brain, so we can think about neuropsychiatric diseases like schizophrenia, like bipolar disease, like autism. For the first time, we have objective measures of when do these diseases start, how far have they progressed, how do they respond to therapy, and maybe most important, we'll be able to take a disease like schizophrenia and divide it into its discrete subtypes, because schizophrenia is going to be many diseases, not just a single type of disease. And I would predict this really is going to transform the whole field of neuropsychiatric studies, because, again, the blood can give you reflections of what's happening in any organ you want to study, the brain, the liver, the kidney, or whatnot. So this I think, is going to be revolutionary and transformational. And I'll tell you, in closing, with a company I started about 4 years ago called Integrated Diagnostics, we've applied these new systems approaches to making blood a window. And we now have a 13-panel biomarker set that allowed us, with enormous accuracy, to distinguish normal lung nodules from cancerous lung nodules. And just to give you a perspective, every year in the U.S., we see 3 million of these nodules, and every physician is faced with the question of, what do I do? Now that we can distinguish the benign from the neoplastic, we estimate that we can save the healthcare system with this one test between $5 and $10 billion a year. So that shows another way of approaching the brain and how you can use the reflections the brain sends out into the body to assess with enormous clarity what's happening in the brain.
I think why this panel is so wonderful is because you see the bandwidth of neuroscience. And I think it's like an orchestra. You bring people together like a cellist, you bring in a violinist, and they're the best in the world, playing together the question how the brain actually works. And so the other thing is that you bring people from outside the field in a way that have spent all their life working on genes, doing nothing with the brain, but suddenly these genes become relevant for the brain, and then they can transform the field. And I think what you heard from Dr. Hood is exactly one of those events where someone who knows proteins like nobody else and genes like nobody else is entering the field of neuroscience and revolutionizing it. And I think we need a lot of this to solve the problem. Now it's kind of my turn a little bit to tell you what we're doing. And at the Brain Research Institute in Seattle Children's. And Christoph just mentioned that what we're doing right now is pouring oil on the car to heal the engine. And in fact, the truth is that the whole brain is full of little engines. So it's not just one engine, but we have an engine for locomotion. We have an engine that wakes us up in the morning. So even if you had a bad sleep in the morning, you get up. And then in the evening you get tired, even though, let's say, you didn't do anything, you get tired because you have an engine that tells you 24 hours to go to bed and get back up. And we have engines throughout the brain, so we're really a rhythmic machine. And everybody's aware that we have to breathe. And we don't want to think about breathing, so we have an engine for breathing. And these engines are not just sitting everywhere in the brain, they're very, very localized. So for breathing, for example, you have an engine of like 500 nerve cells that are critical. And if you damage these 500 nerve cells, you stop breathing and you're dead. But the beauty about this is that you can take this little engine out of the animal, put it in a dish, and the dish piece of tissue will continue to generate breathing as if it would be in the whole animal. Okay, so in fact, when we look at this, we have not only breathing in the dish now, but we have also a sigh or a gasp. So everybody associates like a sigh, like you see a beautiful girl or a beautiful man or whatever background you have, and then you sigh, okay? And then now you have a dish, and the slice will also sigh. And that's a really fascinating question. And having it in a dish allows you now to dissect the mechanism that gives rise to this. So you can now take this engine and take it apart and understand, how do neurons talk to each other? How do proteins actually play a role in setting up this engine and making it work? And so we spend quite a lot of time, so we understand exactly the conditions that lead to a psi. And so you say, okay, well, psi is a pretty romantic problem, but it's not quite romantic. But it's also a really clinical important problem, because most of you are probably not aware that psi is very important for arousal. Okay, so 95% of the time when you wake up at night, you actually wake up with a psi. And everybody who shared a room with someone who snores knows that. So you have these people that snore like, and then the brain gets hypoxic, and the hypoxia means that not enough oxygen will trigger a psi, and you do this, and then you wake up, and then you change your position, and then you start sleeping again, and you can wait 10 minutes later again. Okay. So the psi is like a monitor for not enough oxygen and arouses the rest of the brain. And we know that in children that later died of sudden infant death syndrome, that they did not psi. And so basically they sit on their pillow, they don't breathe enough, and the brain doesn't warn them, doesn't arouse them, and so they can end up dying. So understanding the psi becomes a huge clinical issue. And not only for this, but for sleep apnea, a major problem with obesity. So understanding these engines is a tractable problem. We can take them out, we can dissect them, and we can actually bring it back to the clinic. And this is an interactive process. And now you have to imagine the brain is full of those engines. So we can take out an engine that controls our motivation. Okay, so we have every second you have to make a decision to do this or not to do it. Okay, so do I want to drink a beer now? No, not a good idea. So I say, not a good idea. And so I have this stop pathway that tells me not to do it. Some people, however, don't have that. They're very impulsive. They have ADHD, or they have other Tourette syndrome where they have to do a movement. They cannot stop that movement. Or they have OCD where they start to wash their hand. They cannot stop that movement. Or they have the opposite. They have Parkinson. They can't initiate a movement, so they are very rigid. And so all of this involves the same circuitry, the same engine. And we can take it out and put it in a dish and understand the genes that give rise to this onset and offset. And we can relate it to psychiatric disorders. And we can do the same thing for engines that control the temporal processing in our brain. So in order to have social communications, you have to observe the face of a person, how it changes. You have to observe not only the face of a person, but you have to observe the movement. And all this is going on in a very, very rapid way. And you have to have a temporal processing center in your brain, an engine that sets up the timing. And this is sitting in the inferior olive. And if you have a problem with this circuitry, then you have problems with temporal processing. And we know that a lot of children with autism have this problem. They cannot process things that are faster than 300 milliseconds. So what they can't do is they can't communicate with other people because it's way too much for them. So they can focus on things that don't require so much adaptability, fast processing. But the tragedy is this is only one aspect of autism. There are many, many different forms of autism. And what we have to find is a synthesis of all of us here, sitting here. We have to understand the proteins that are important for different forms of autism. And then we have to have these engine people that can dissect, what are these proteins actually doing to this engine in order to understand how we can actually interfere with this. And then we have to understand the cortex people, because a lot of these things, the psychiatric disorders, are really problems with, the cognitive function in the forehead versus the emotional functions in your body, which are sitting deeper inside in the brainstem. So I think what's so fascinating about neuroscience is not a single person can do it. You have to have these car mechanics that understand engines, like myself. You have to have the people that understand the fluids and the oils of the machinery. And you have to understand the people that work on the cognitive function, on the steering wheel of your car. And then you have to also have people that understand the real problem, and that's where Pete comes in. And I think this is kind of the task of the future, is to bring everybody together so we are not working in silos. And I think we've worked for too many years in silos. And I have to tell you that even though we're sitting in the same city, what Lee just told you is the first time I've heard about it. So this is impossible. And so we have to do something about it. And knowing Pete, maybe Pete, you want to tell about what do we have to do to solve the whole problem now?
Well, I'll do it very you asked a non-neuroscientist to explain what we have to do to solve the problem. Well, first of all, if you just think of what Dr. Hood just told you, I mean, think about that. I mean, we would have an actual diagnostic tool where we know somebody had post-traumatic stress. Instead, today we have 14 questions in a psychiatric manual called DSM-4, soon to be DSM-5. And basically, it can be interpreted different by psychiatrists, just like what happened down at Fort Lewis not too long ago. Two different sets of psychiatrists interpreted the answers to the questions in different ways and came up with different diagnoses. But just think of what he said with traumatic brain injury. Well, a very interesting thing what happened down at Fort Lewis. There were more folks down at Fort Lewis who had an initial diagnosis of personality disorder that were given a diagnosis of post-traumatic stress than there were reversed the other way. That didn't make it into the newspapers. I won't go there. What Dr. Hood said about taking the idea of a blood biomarker to concussion, if we could really find a blood biomarker that would give us with certainty or close to certainty that an individual had a concussion, wouldn't that be wonderful to have an assay machine, not unlike what a diabetic uses to look at his blood sugar, that you could take on the sidelines of a football game or a girls' soccer game or wherever, and with a simple prick of the finger, be able to tell with some form of certainty, up to possibly 95% or maybe even higher, whether or not that individual actually had a concussion or not. And wouldn't it be wonderful if we could have that? And that's what I find so exciting about this. But when you're around neuroscientists, they always say the heart only exists to pump blood to the brain. It is the brain that makes us who we are. And it is, as has been explained today, the toughest of all problems. And I believe the only way we are going to get at it is to break down the silos and get people working together to share data, to collect data, to make sure that the scientist who collects data in Alzheimer's is sharing it with the scientist who is working on ALS or Parkinson's. That, to me, is what you're going to have to do to understand this most complex of organs in our body. It's not going to be solved by scientists at Stanford or Yale or Harvard or any one place. It is going to be solved when we get people to work together and create a business model that allows that to happen, where data can be shared and people who go down a dead-end alley tell everybody else they went down a dead-end alley so people don't follow them down that same alley, as happens so often today. And with that, I will stop, and I think we've opened it up to questions.
Let me make one comment before we close, and that is an extension of Pete's comments. We are on April 14th and 15th having a big symposium at the Institute of Systems Biology on the brain. And some of the people you've seen up here, in addition, many others will be there. So if you'd like to extend your education, this is really a unique opportunity where we bring together the best and the brightest from all over the world, really. Okay.
And I want to emphasize again why this is a revolutionary idea. And university were set up in departments. You have a physiology department, you had a genetics department, a molecular biology department. And in the molecular biology department, you have some people working on the molecular biology of the brain. You have the genetics department, a geneticist working on the brain, and you have a physiology department, and physiologists working on the brain. But they don't see each other because they're sitting in different buildings. And I think what we need to do is break those silos, bring them together. Allen Institute is one example. Our Brain Research Institute is another example. And there are many of those examples coming up. But it's a revolution, and I think that is what we need. And so it's not just money. It's a different attitude towards the brain. that the brain is not solvable by individual, it's solvable as a team. And we've learned this from the other systems, the other projects, the big science project, and I really think it's time for neuroscience. So with this, I would like to open this for question and answers, and we have microscopes, no. And I think there's a microphone and there's a microphone, and that makes it also easier than say you over there or there, so we go one way or the other.
And it would be nice to say your name, too.
Yes, nice to say the name, and then we'll go for it. Thank you very much.
I am Dr. Kayla Wiener. And Dr. Hood, I'd like to address my question to you concerning PTSD. I'm wondering if in being able to trace the progression, that there's any part of it that you can tell who is likely to suffer from PTSD and who might not. I mean, when you talk about normals and not normals, I mean, I'm not sure anybody comes back from war normal, but in terms of PTSD, is there something that you found or would look for to see who might have a predisposition?
That's really a terrific question. I think people who are experts in this area say unequivocally there are individuals that are utterly resistant to PTSD. There are individuals that are incredibly susceptible. So we hope in time to be able to analyze and compare the genome sequences of those individuals to look for regions that might correlate with this predisposition. But I think and that's a really hard problem, and that may take a few years to do. But I think even more exciting is the idea with these blood markers, what we can do to Army troops before they ever get disposed out to war zones and all, is give them tests that will stress them but won't cause the full-blown syndrome. And in this way, I mean, you could probably identify individuals who should never be Rangers or SEALs. And even more, you can identify individuals who should never, ever go to war. So I think we are going to be able to do that. And in the near term, I think we can use the diagnostics to assess populations that are really at risk. And then later, we'll be able to identify the genes that are involved. And then there's always the hope that one could create therapies that could ameliorate those predispositions.
I would like to follow up on this insofar as this is really a very big general issue, not only for PTSD and post-traumatic stress syndrome, but it's really the case for all neurological disorders. And let's look at ADHD. There is not a single gene that causes ADHD. It's basically a predisposition. You have a number of genes that make you predisposed, and then this kid in this certain environment will develop ADHD. Maybe in another environment that kid would have not developed ADHD. So what we really need to understand is, what are these predispositioning genes that actually then tip us over to develop autism, to develop epilepsy, etc. It's really a general question that we need to address, and this is again the need to bring people together with genetic background, with the physiology and neural networks and cognitive functions. So.
And I would argue that there are people predisposed to concussion.
Yeah.
I think that a lot of folks, a lot of scientists are telling us that genetic makeup has a role to play in concussion. That two people can be hit exactly in the same place with the same force on their head, and one may have a concussion and one may not have a concussion. I find it, and if you notice, there's one little difference in the way we talk. I leave off the D. And let's forget soldiers for a second. We call this a disorder. Is it a disorder or is it an injury? Are some people injured and other people aren't injured? And I would submit to you that 72 to 74% of the women that are sexually assaulted get some amount of post-traumatic stress. That's where most of the research has been done. Now, is it right to go to a woman who's been sexually assaulted and say, because she has that kind of reaction to a horrible event like that she has a disorder? Is that a disorder or is that an injury? And I really feel that there are a lot of folks out there that are not getting the help that they need because a 19-year-old kid, or no one, really wants to be told they have a disorder. And until we know for sure it's a disorder, I think we're doing a great disservice by calling it a disorder. I've made that argument to the APA.
And again, I would like to expand on this insofar as we can go also with ADHD. You know, some children are developmentally a little bit later, and they have these attention deficits or so. But is it a disorder already? In some children, clearly it's a disorder, but in others, it's a symptom of an environmental condition or something like this, because they were not ready for this. And so there's a fine line between, you know, calling it a disorder to calling it a symptom. You know, and we have to learn this. And the same thing is, anyway. Yeah, we can go on. Next question, please.
I would be interested in hearing your thoughts, particularly if you've heard of the work done in the book Blank Slate by Steven Pinker. And he described where they separated the left and the right half of the brain of an epileptic person. And he posits in the book that there is a machine in our brain that basically is a self-justification machine. It makes up stories. He calls it the baloney factory. And my favorite theory has been that the brain is a self-justification machine. That the heart may exist to give blood to the brain, but the brain exists to tell us that whatever we want to do is what we should be doing. But so I would just be interested in hearing your thoughts on that idea, particularly Steve Pinker's idea.
Yeah, so these experiments were done at a place where he was the chairman of for many years at Caltech, a so-called split brain experiment done by Roger Sperry, who received the Nobel Prize for them. And then what people discovered that typically in most people you have a dominant and non-dominant hemisphere. Typically in most people the dominant one is the left, that's the one that talks, or the right hemisphere typically is less eloquent, it can sing, it can answer very simple question, but by and large it doesn't have the language capability that typically most people is on the left. Then people like Pinker, particularly Michael Zanica, have then built sort of more elaborate cognitive science theory on that, saying one part of the brain sort of is more sort of self-justifying than the other. I'm not sure. I mean, the evidence for that is much less compelling than the evidence for very strong lateralization of language. That's really the best evidence we have that the left brain is different from the right brain. If you look at the genome expression and you look at the architecture of the brain, it's actually not easy to see differences. So we have these brain atlases at the Arm Institute. We spend considerable effort to look for differences between the left human brain, the right human brain at the level, for example, of gene expression, which genes are expressed, and you really can't find any. That's not to say that there aren't some there, but they don't seem to be very pronounced. So I think there's a lot of sort of law about the left brain and the right brain, and claim some people are more left brain, other people are more right. I think it's a lot of just so stories rather than real solid science.
Hi, I'm Deb Frickett, I'm an architect. Just a science groupie. I've been reading in the press about epigenetics as related to cancer. It's called the biggest revolution since it was first understood that cancer was genetic to begin with. I'm just wondering if epigenetics comes into play with your work. Any efficacy, any directions that'll take you in?
Okay, can I just but I'm not the expert, but just one concrete example is, for example, sleep apnea, and you start having an obstruction, and so you don't have enough oxygen, and then you have intermittent hypoxia, so you get problems with sleeping, etc. But you get also huge cognitive problems, and you change blood pressure problems. And even if you heal the obstruction, you will still have blood pressure problems for the rest of your life, and it becomes ingrained in this. And now we know that a lot of the epigenetic mechanisms are turning on and change your whole system for a permanent way. And so understanding basically how epigenetics plays a role in determining the long-term outcomes of disorders is a huge issue right now, and a big topic of investigation. So I would just say epigenetics, from a non-expert,
is the ability to modify the DNA so it alters its pattern of expression. So you basically can turn off big chunks of information. And what's interesting about epigenetics is it can be inherited from parents to children. So these traits that turn off certain segments of the genome can actually be passed on. And at a later stage, these marks that turn them off can be removed. So it's a thermostat for the genome that we never knew existed before. And it's an enormously voguish, popular area that is involved in almost every aspect of biology today, one way or another. I think it's slightly overblown, but it's really impressive what it can do.
I have seen a scientific presentation at Neuroscience conference where they exposed a female mouse to nicotine, and then the offsprings were hyperactive. And this hyperactivity lasted for 4 generations after this. So 4 generations after this, the mother that smoked had an hyperactive kid. And so this is... And interestingly, it was for the female, and the male was not the big issue here.
That is really important is these epigenetic phenomena can occur very early in childhood. So early childhood experiences can be seminal for the rest of your life. So, anyway. Next question.
Hello, my name's Joe Brewer, and I'm an independent interdisciplinary scholar. And I do work applying cognitive science to social change to deal with the global ecological crisis and the political and economic systems. And I do not have a PhD because the university is not really set up for interdisciplinary research. So the question that I have, considering that we all agree here that this research is incredibly important, as is Earth system science and the ecological crisis, which, by the way, is deeply relevant and interdependent with brains research. How do we go from the siloed model of academic research and non-academic research in its various forms to the models that truly embrace interdisciplinary, synergistic, holistic research, so that the literally thousands of people that I personally know who no longer are in academia and did not pursue a PhD and are working on these problems can be re-engaged, and so the next generation can participate?
Well, I can give you a really simple answer to that. In the year 2000, I resigned from the University of Washington to start an independent institute to do systems biology and so forth. And the principal reason I did was the bureaucracy at state universities doesn't permit you to do innovative, out-of-the-box, original thinking, and it perpetuates the modes of doing science that previously existed. I had a cross-disciplinary department of biology, and I'd hired this spectacular young surface chemist from Penn State, and the dean of the medical school walked into my office right after I made this announcement and said, "I'm unconvinced a surface chemist has anything to do with medicine." So I gave him my best argument, and he said, "I'm totally unconvinced." And he said, "I'm not going to release the money to do this kind of thing." So I think for new ideas, it takes new organizational structures, one way or another. And I think the Allen Brain Institute is an example. And our institute are examples of institutes that are flexible, they're small, they can make decisions incredibly rapidly, and they're very attractive to scientists that are entrepreneurial and fearless and want to explore the future.
Christoph is clearly also an example from Caltech to here.
Leaving my tenured position next week.
So you have to save all of us if Obama doesn't give us the money.
Well, it must be in the water at Pasadena because I actually came from Caltech as well, somewhat earlier.
Hi, I'm Dennis Fortinos. I'm actually Canadian. I'm here visiting some friends who brought me here tonight, and I'm very thankful. And I hope Obama does give you the money and we benefit in Canada as well. Being Canadian, I'm fascinated about concussions, and you talked about concussions. My son plays hockey, and of course we have a passion for hockey, and in particular, my fascination is about consciousness and how it relates to concussions. Recently, I'm not sure if you heard the story here in the U.S., but a Russian hockey player received an open ice body check, fell to the ground, got up, hobbled off the ice, skated off, actually came back on, had 2 or 3 more shifts, finished the game, went out drinking. The next morning he was dead in his bed. And they said it was a concussion. My son plays hockey, so it's very dear to me, and he's in his NHL draft year this year. Last year's season was cut short for 6 weeks because he's a goalie, and he got a puck to the chin, and he was out. But interestingly enough, he had to get hit twice in the chin, and only after the second time, after they told him, we think you have a concussion, did it seem to get worse. So the question I have is there some kind of relation or correlation between some kind of cognitive ability or consciousness or awareness to concussion? Because before he was told he had a concussion, he continued to play and felt he didn't have any symptoms. The moment he was told he had a concussion, he started the brightness of the ice and lights and all the symptoms that go along with concussion seem to come forward. So is there some kind of work and correlation on that?
That's a great question. I can't answer it.
I think what we have to realize is that concussion is not concussion. It really depends on what areas in the brain are affected during each kind of blow. And we're just starting to learn this. I mean, like one of the big revealers in the soldiers was that in fact it's not necessarily the blow that you get on the head, but the blow that you get on your stomach that pushes up the cerebrospinal fluid that then changes internally your cortex is a big deal. And so we're really understanding, just in the process of understanding, really, I mean, like the differential effect of a blow. And it depends on where you're hit, what kind of symptoms you will have. And that's, in the brain, location is always the key thing.
You might be one to ask the question, did he have a concussion the first time? What we saw in soldiers, and I think that's why you see some of the rules that you see in the NFL today, that if a person receives a second concussion before the first one had an opportunity to heal, that's when we saw the cognitive issues and the symptoms that occur that cause some real difficult problems. So rather than look and maybe attribute that, and again, this is the non-doctor speaking, but what we saw was that it's that second blow where the symptoms can be much more pronounced. At least that's what we saw in soldiers, and it was with the first, if the head injury had not had an opportunity to heal.
I think this again addresses a very fundamental problem, fundamental issue in neuroscience, and I think it was also addressed in the previous question about the self-regulatory processes of the brain. And we have only 30,000 genes that set up like 100 billion nerve cells with very precise connectivity. So the 30,000 genes cannot tell exactly what neuron is connected with what neuron. So this has to all work by a homeostatic regulatory mechanism, so self-regulatory mechanism. And these are playing a role all the time in our brain all the time. So a comparison is if we fly with a jumbo jet from here to Europe, it's an 8-hour flight, let's say 8-hour flight. By the time you arrive there, every little part of the Boeing has been replaced, because all the time you have turnover of proteins and regulatory process. And so you have to have all these homeostatic mechanisms, and yet we are not all crazy, because these homeostatic mechanisms are working. So let's say you have a concussion the first time. The brain starts a process of adjusting to your concussion. So you start a homeostatic process that tries to adjust to this concussion. But now your brain is not the same anymore. So now comes the next blow. And so now it becomes vulnerable, because now you shifted your homeostasis into a different direction. I think one of the ideas we think why neurological disorders are so difficult to cure is because of those homeostatic regulatory mechanisms. So basically, if you have an epilepsy, yeah, you can give anti-epileptic drug, you can reduce the excitability in the brain for a while, but these homeostatic mechanisms are working, and after a while, again, you have an epileptic seizure. So I think these whole self-regulatory processes are very fundamental for understanding traumatic brain injury, for understanding, but also neurological disorders in general.
I'll tell you, another point that I make is until we have a simple genetic test that can determine whether you're susceptible to traumatic brain injury or not, I would never let a son or a daughter play football. I would really question soccer. I would really question hockey. I think you ought to think very, very seriously about these things, because some of these concussions have lifelong effects, for particularly, presumably, for people that are susceptible, and we just don't know who those people are going to be right now.
What about biking and skiing and driving?
Skiing, don't take away skiing.
Until we get such a test, we can't do any of those.
No, I would say if you look at how many times you get hit during a football game, it's an incredible number. And the other things are occasional, rare kinds of accidents. So I wouldn't put those in the same category. But you know, people have argued that hitting a soccer ball can be equivalent to the kind of experience a football player gets. So you really have to think about these things, especially if they're your kids, right?
Well, we had a party where my brain research friends came, and then one of my colleagues came, and the kid was 3 years old, coming with a helmet. So so there's a balance we have to find. I didn't name it.
Thank you. I'm a nurse practitioner. I was wondering, I was glad to hear that like 5% of the research on the Human Genome Project went to ethical issues. And I was wondering about a couple of issues. One was like the breast cancer gene that was patented and is very expensive because of that, and how that happened if it was really more open source. And also, often science can precede the ethical things, and what happens if you have a genetic test or can test something and you don't know exactly what it means, and then it's like if somebody says, well, now you can't go into the service to serve the country because you're too high at risk, and how sometimes that might shut doors, and how you anticipate maybe dealing with that.
Well, I think the issue of being able to diagnose predisposition to a disease without being able to do anything about it really does present a conundrum. And there are a number of different ways that you can handle it. One is you can give people a right only to know the good things and not to know any of the bad things. But as we move toward an era when all of you are going to have your genome sequenced, and there are really compelling reasons for doing that, what is going to be absolutely essential is passing laws that prevent discrimination based on employment, on insurance companies, frankly, on the basis of your own family in some cases. So I think these discrimination laws, anti-discrimination laws, are really, really going to be important. But I think what's going to be a real driver for the Human Genome Project is the fact now that we've identified more than 300 what I call actionable variants. So an actionable variant is a gene variant which, if you have, I can tell you something that's going to improve your health unequivocally. And an example is a friend of mine in Microsoft who started getting osteoporosis in his late 30s. He was lucky enough to have a gene analysis of just a few genes, but they found he had a defect in a major calcium transporter, and he totally reversed his condition in a year and a half by taking 20 times the normal amount of calcium. So there are 300 genes like that. Wouldn't you like to know if you had one or more of those and you could change your future health by treating? And as we go on, there'll be more and more of these. So once you have your genome done, you've invested in improving your health for the rest of your life, because we'll check every year to see if there are new options we've identified that may be present in your genome where we can deal with them effectively. But the whole issue of genes that may predispose to disease that we can do nothing about, it's a real conundrum. And my feeling is, I think people should decide whether they want to know if you have an ApoE4 gene, which has a low tendency to predispose to Alzheimer's or not. So those are real questions that different people answer differently.
Yeah, and it's very critical now, for example, in cancer therapy. I mean, basically, if you know exactly what kind of genetic background you have that caused, let's say, this tumor in, let's say, breast cancer, it really tells you exactly what is the medication that helps you best. So I think there's a lot of benefit, and at the same time, there's a lot of ethical issues. But I mean, we all have to go through IRBs and IACOOCs, and it's an issue. Yeah.
Let me just say one more thing. You asked about the 2 breast cancer genes that have been patented. My own view is, as we move to complete genome sequencing, that'll all disappear, because everybody's going to be identifying those genes, and there's no way that company can go and sue every one of you. So I think once we democratize the information, the ability to have patents will disappear.
My name is Howard Niebeck, and I was wondering if some of this impending research will be directed toward learning, or understanding how we learn, and in particular, what's going on with learning disabilities.
Most of the money, if you mean the Obama Brain Initiative, most of that money is going to be dedicated to for neurotechnologies, to study the brain, to develop devices to probe the brain in ever finer resolution, to probe, to design devices that can record from larger parts of the brain, and devices and genetic techniques to interfere with the brain very selectively and very specifically, so that you'll be able to turn on or off specific groups of cells using different colored lights. It's a fantastic, it's almost a magical technology. And with some of those tools, they'll study learning. But most of the money is going to be for technology for basic research.
But I mean,
in the Obama initiative.
Exactly. But the question how learning works in the brain has a long tradition in neuroscience, and I think one of the interesting new developments of this whole question was that, you know, when I was going to grad school, we learned that, you know, you're born with a number of nerve cells, and they are not new nerve cells. And now we know that basically there are areas in the hippocampus that is involved in learning where nerve cells are born. And what we also learned is that actually through activity, bicycling or running, etc., you actually generate more of those nerve cells. You know, like so. So now they are generated more of those progenitor cells, but it doesn't mean a lot if you don't do any cognitive work. But if you do some interesting work, then actually these neuron cells get actually integrated into your brain and become part of the learning circuitry. So there's a lot of plasticity that has to do with activity and cognitive work, etc. So I think we know a lot about this, and what we need to do is apply this to our behaviors, etc. And I think Christoph biking here is one of the reasons that he wants to have more progenitor cells.
but I would just be careful. And here's where I'd be careful. I would be careful. I quoted $78 billion for traumatic brain injury every year, and we're spending $84 million. We had PricewaterhouseCoopers, because what we're trying to do is wrap our arms around all the diseases of the brain. We had PricewaterhouseCoopers go out and do an economic analysis for us on what we pay if we were to combine all the diseases of the brain together, yearly cost. And the same thing happened in Europe, $1 trillion a year. Each place, $1 trillion a year. That's $2 trillion. We're putting $100 million against a $2 trillion problem. Now, I'm an optimist. You have to be an extreme optimist to think that that's going to get at that problem.
Well, on the other hand, we have already, we have a National Institute of Health. The budget of the NIH is $31 billion. So, that's not an inconsequential sum of money. We have a National Science Foundation whose budget is $5 billion. So, as a country, we're investing, on the order of $60 billion in one form or another.
But the $31 billion is not going against the brain.
No, it of course includes cancer and other things, but the total money that's being spent by the federal government is much, much bigger than what was announced yesterday.
And philanthropy is a big deal. That's where the Allen Institute comes in, where people believe in it and actually give all their fortunes into something that they believe and informally. And I think this made this country really like where we are right now.
We will collect, we have collected.
$5.
And there are 3 different colors for each of the institutions. We had dinner last night with the former Minister of Economy of Luxembourg, and he really marveled at philanthropy in the States, because in Europe there is nothing comparable whatsoever. And I first knew him about 6 years ago, and he was absolutely determined to get through at least the first of the initiatives that would bring to Luxembourg at least the early stages of philanthropy. And he just barely got started. It is an enormous struggle, and the real struggle isn't the laws. It's the entrepreneurial people in Europe who want to spend the money on themselves and their families and their curiosity. It's a culture. And here we have just a terrific philanthropic culture. Very different.
I just want to jump in really quickly. Thank you all so much for coming. We have time for one more question. So thank you all for your thorough answers and for your interesting questions, but we're going to just take one more. Thanks so much.
Hello, my name is Charmaine. First off, I just really wanted to thank all of you for being here. I feel like I'm in the presence of superstars. I've read about all of you. My question is for Dr. Christoph about that. One of the goals of the Allen Institute, I think, is to unravel unconsciousness, as you mentioned. My first question is, how do you define consciousness? And my second question is, so I've done I'm a neuroscience PhD student, so during my neuroscience research, what I found is that what we tend to study is different, physical aspects of the brain. As in, does this synapse connect with this synapse, or does this protein exist in this part of the brain? Do you think that in our lifetime we'll be able to transcend from going from the physical, figuring out the different genes and their expression patterns and the connectomes, etc., to actually be able to understand how that turns into consciousness?
Yes, and we're doing it every day in the lab. So, for instance, you can doing brain surgery that happen, right? A surgeon stimulates, and you can also do it experimentally now, stimulates a particular part of the brain, and you have a conscious experience. You see red, or you might remember, a very specific memory. So here you have, if you want, the water of the brain turning into the wine of consciousness, right? So here, the mind ultimately is what the brain does, right? The mind is a manifestation of the brain, and the conscious mind is a particular manifestation of certain parts of the brain. They're linked, they're twins, they're just the different sides of the coin. And the challenge is to exactly understand the laws, the bridging principle that link specific neural activity with specific conscious perception, but people, we are doing this now. We and other people are doing this in the lab. We don't have to wait for some mysterious point in the future when we'll know it. And the central challenge still remains to explain how the physics of the brain, after all, the brain is a physical system like black holes, like viruses, how the physics of the brain gives rise to subjective feelings. And to explain, for example, why these guys, because they also have a physics, right? They're also very complex, while these guys, at least we think right now, do not have conscious sensation. So that's what people are trying to do. And I think, yes, in our lifetime, we will see sort of explanation that most people, at least most scientists, are going to be quite comfortable with.
And anesthesia is also not a
well, they have to turn it off.
Where they can turn it off. So where exactly is anesthesia hitting? So you lose your consciousness. So these are with all these gene tools where you can knock out certain areas and see suddenly your anesthesia sensitivity is gone. So I think there are mechanistic ways to tackle it. Demystifying the brain, that's what we